You're eating chia seeds wrong. And I don't mean the recipe, I mean the science. Because there is a step almost no one takes, and skipping it means you're throwing away the most valuable thing in that tiny seed. I'm going to show you what happens inside your body when you soak chia seeds versus when you don't, and why it could be the difference between feeding your fat loss bacteria or starving them. If you're over 40, eating clean, doing everything right, and the scale still isn't moving, this is not a willpower problem, it's a gut problem. Here's what the research is showing us. The balance of bacteria in your large intestine directly affects how your body handles fat storage, hunger, and insulin sensitivity. And one bacterium in particular has become one of the most studied microbes in metabolic health science. It's called Akkermansia muciniphila. Studies consistently show that people with higher levels of this bacterium tend to have lower body weight, better insulin sensitivity, and less visceral fat. The problem? Most of us, especially after 40, have far too little of it. Here's what most people miss. The food you eat doesn't just fuel you, it fuels them. So what does soaking chia seeds have to do with any of this? Everything. Stay with me. When chia seeds hit water, they release a clear, gel-like coating called mucilage. Most people see that gel and think it's weird. What they don't realize is that gel is a prebiotic fiber, a direct food source for your gut bacteria. It travels through your stomach and small intestine untouched. Then it arrives in your colon, where your bacteria fermented and produce the metabolic signals that may influence how your body stores or burns fat. But here's the important part. If you eat your chia seeds dry, the gel forms late, forms incompletely, and your bacteria get a fraction of what they should. Soaking changes the equation entirely. Chia seeds are about 38% fiber by weight. Roughly a third of that is soluble. When the seed absorbs water, it forms a thick gel. That gel is the mucilage. In simple terms, think of a dry sponge versus a wet sponge. Dry chia expands in your gut, but slowly and unevenly. Soaked chia arrives at your colon as a fully activated sponge, ready to feed exactly the right bacteria. Chemically, chia mucilage is a polysaccharide, a long chain of complex sugars your digestive enzymes cannot break down. Your stomach cannot dissolve it. Your small intestine passes it through completely intact. It arrives in the colon like a delivery package addressed specifically to your gut bacteria. This is where it gets interesting. Not all fibers feed the same bacteria. Research published in the Journal of Functional Foods used a dynamic model of the human gastrointestinal system to study how chia mucilage interacts with gut bacteria. The findings suggest that the branching structure of chia's polysaccharide chains may selectively fuel specific beneficial strains, including those linked to metabolic health. A 2025 study in Food Science and Nutrition compared chia and flaxseed fiber fermentation in the colon. Chia increased the abundance of roseburia, a known butyrate producer, and other strains associated with reduced obesity risk. In other words, chia mucilage doesn't just feed any bacteria. It appears to preferentially feed the ones that matter most for your metabolism. And the most important of those is Akkermansia. Akkermansia muciniphila was first isolated from the human gut in 2004. Since then, it's become one of the most intensively studied bacteria in obesity and diabetes research. It lives in the mucus layer of your intestine, that thin, gel-like lining between your gut wall and the bacteria in your colon. Its job is to feed on mucin, the protein that makes up that mucus layer, and in doing so, it signals your gut to produce more of it. In simple terms, think of Akkermansia as the maintenance crew for your gut lining. When the crew is well staffed, your gut barrier stays tight and sealed. When the crew is depleted, gaps form. And those gaps are dangerous. When your gut lining develops those gaps, something that happens with age, poor diet, chronic stress, and low fiber intake, bacterial toxins called lipopolysaccharides, or LPS, leak into your bloodstream. This is called metabolic endotoxemia, and research shows it is a primary driver of systemic inflammation and insulin resistance. Here's why that matters for your fat loss specifically. Insulin is the hormone that tells your fat cells to release stored fat. Think of insulin as a key that unlocks your fat cells. When your cells stop responding properly to that key because inflammation has jammed the lock, fat burning slows, fat storage accelerates. You can be doing everything right and still be fighting your own biology. Now here's where the science gets genuinely exciting. A randomized, double-blind, placebo-controlled pilot study published in Nature Medicine supplemented overweight, insulin-resistant adults with pasteurized Akkermansia muciniphila bacteria over 3 months. Compared to the placebo group, the Akkermansia group showed a 28% improvement in insulin sensitivity and a significant reduction in blood insulin levels. They also saw modest reductions in body weight, fat mass, and hip circumference. These are modest but meaningful results from a pilot study in 32 participants. The researchers themselves described the effects as preliminary. This is not a miracle treatment, but for people who are metabolically stuck, and many over 40 are, improving Akkermansia levels is a strategy worth understanding. The question is, how do you support it through food? Akkermansia is what scientists call a mucin-degrading bacterium. It feeds primarily on the mucus lining of your gut. But it's also highly responsive to the broader fiber environment in your colon. When prebiotic fibers are present, other bacteria break them down into smaller fragments. Those fragments support Akkermansia's environment. In return, Akkermansia produces compounds that support those bacteria. It's a cross-feeding loop, each microbe helping the others thrive. In simple terms, feeding Akkermansia directly is hard, but feeding the ecosystem around it? That's something your diet can do every single day. But here's the important part. Not every fiber does this equally well. A 2022 review published in Food Frontiers analyzed how different dietary fibers affect Akkermansia abundance in the gut. Soluble fibers, particularly those from seeds and plant polysaccharides, consistently showed the ability to support conditions where Akkermansia flourishes. A landmark animal study published in PNAS demonstrated that prebiotic supplementation in obese mice normalized Akkermansia levels, and that restoration correlated with improved gut barrier function and metabolic markers. The researchers observed that Akkermansia dropped dramatically in diet-induced obesity, and feeding the ecosystem through dietary fiber helped reverse several of those metabolic changes. For you, eating soaked chia seeds means delivering a fully bioavailable prebiotic payload to the exact environment where Akkermansia lives and works. You're not just eating fiber, you're replenishing the maintenance crew. Now let's talk about what actually happens when gut bacteria ferment chia mucilage. The end products are called short-chain fatty acids, SCFAs. The 3 main ones, butyrate, propionate, and acetate. These are not waste products. They are functional metabolic signals your body uses in powerful ways. Here's what most people miss. Your gut bacteria aren't just digesting your food, they're sending hormonal signals to the rest of your body. Butyrate. This is the preferred fuel for the cells lining your colon. It strengthens your gut wall, reduces local inflammation, and helps maintain the tight barrier that keeps LPS from leaking into your bloodstream. Less LPS leakage means less inflammation. Less inflammation means insulin works better. In simple terms, butyrate is the repair cruise fuel. Without it, the gut wall weakens. With it, the wall stays sealed and your metabolism stays calmer. Propionate. This is where things connect directly to fat loss. Propionate travels through the gut wall and signals specialized cells to release GLP-1 and peptide YY, two satiety hormones. GLP-1 is the same hormone pathway targeted by weight loss medications like semaglutide. When your gut bacteria produce propionate naturally through fiber fermentation, they activate a quieter version of that same signal. You feel fuller, you eat less, without the pharmaceutical price tag. A 2025 systematic review in the MDPI journal found that Akkermansia muciniphila is among the major contributors to acetate and propionate production in the gut. The review also noted that SCFAs can influence gene expression in fat tissue in ways that may favor fat oxidation, though most of this evidence is still from animal models and human trials are ongoing. Dr. Jason Fung's clinical work with metabolic patients has long emphasized that gut health, insulin resistance, and fat accumulation are deeply connected. His clinical observations align with what this emerging science is showing at the molecular level. Now let's put the full chain together. This is what happens when you eat properly soaked chia seeds. The mucilage forms fully during soaking. It reaches your colon intact, untouched by your upper digestive system. Gut bacteria ferment it, producing butyrate, propionate, and acetate. Propionate signals your gut to release GLP-1 and PYY. You feel fuller longer. Butyrate strengthens your gut wall, blocking LPS and reducing inflammation. Reduced inflammation supports better insulin signaling. Better insulin signaling means your fat cells are less locked in storage mode. This is not a miracle. Dr. Christopher Gardner's research at Stanford has consistently shown that individual responses to dietary changes vary widely based on your baseline microbiome, your genetics, and your existing metabolic health. Some people will feel this shift in weeks. Others will need months of consistent fiber intake before the microbiome responds. But if your fat loss has stalled, if you're over 40 and metabolically sluggish, and if you've tried calorie restriction without lasting results, this is a lever your body actually has, and it costs you nothing but 30 minutes of soaking time. Here's the real takeaway from all of this. The gel that forms when chia seeds soak in water, that thing you might have ignored or even squeezed out of the jar, is not a cosmetic feature of the seed. It is the functional part. It is the prebiotic that your gut bacteria actually use. Without soaking, the gel forms late, incompletely, and your colon gets a fraction of what it should. With soaking, you deliver a fully activated prebiotic directly to the bacteria responsible for your gut barrier, your satiety hormones, and your insulin sensitivity. 20 to 30 minutes, that's all it takes. Your metabolism isn't broken. Your gut bacteria are waiting for the right fuel. And understanding that gives you something real to work with. Not a promise, but a mechanism. If this shifted the way you think about how you eat, not just what you eat, then give this video a like. It genuinely helps more people find this content. Drop a comment below. Tell me, did you know soaking mattered before today? Are you going to start? Type yes in the comments if you're committing to soaked chia for the next 2 weeks. I read every one of those. Share this with someone over 40 who's frustrated with stubborn fat and deserves better information than just eat less. Subscribe for evidence-based content on gut health, metabolism, and fat loss. No hype, no false promises, no quick fixes. Just what the science actually shows.